Hey guys, Michael and I are doing our grant proposal on the effects of common goldfish, Carassius auratus, on native aquatic plant density and species richness. So invasive species can be detrimental to new habitats and can have many unexpected impacts on native plant populations. Goldfish are a popular species in the family of carp, commonly kept in aquaria and often released into the wild once they're too large to care for. In the 1800s, they were deliberately stocked in ponds to meet the demands of ornamental aquaculture, and it was quickly recognized that they can become invasive to the ecosystems they inhabit. Native aquatic plants are important for maintaining water quality, food, and shelter for various species across the trophic levels, which is why it's important to monitor and protect them. The aim of this study is to determine how goldfish affect species richness and density of native vegetation after introduction. The study will take place in two locations. The first is Lake Tahoe Basin, California, where goldfish were introduced by aquarium owners as far back as 1862 and still remain in high abundance. And the second study site is Pyramid Lake in Nevada, where no goldfish are currently living. These two study sites will function as high and low levels of goldfish abundance. These sites are ideal because they are ecologically similar and have identical plant species. We can compare how goldfish have impacted native flora in Lake Tahoe by using Pyramid Lake as a control, since there are no goldfish present. Some of the native species we expect to see in the study are Andean milfoil, native elodia, coontail, and pondweeds. We hypothesize that having goldfish present will negatively impact native plant density and species richness. We predict that both native plant density and species richness will be lower in Lake Tahoe than in Pyramid Lake due to the presence of goldfish in Lake Tahoe. The relationship between goldfish and native plants will be further tested in a lab manipulation experiment at the Sierra Nevada University near the field study sites, where the same native plants will be observed in a controlled setting. Fish tanks containing a representable abundance of goldfish, which will be determined from the initial sampling of lakes, will represent lakes with goldfish present. Fish tanks with no goldfish present will be used as our control. From this experiment, we can measure the direct effects of goldfish's herbivorous behavior on the native plant species richness and densities. So as I mentioned, our field methods will take place in two study sites, Lake Tahoe and Pyramid Lake, chosen because goldfish were introduced to Lake Tahoe in 1962 or earlier. That has resulted in massive goldfish being caught by fishermen today. This lake also has other invasive fish present, such as brook trout, rainbow trout, bluegill, and brown bullhead, which is why we're also carrying out the lab experiment that excludes other fish that could possibly predate on plants. And then Pyramid Lake was chosen as a control because it has no goldfish present and is extremely similar to Lake Tahoe because it's actually hydraulically connected to Tahoe by Truckee River, which drains from Lake Tahoe. Truckee River has no reports of goldfish present and is currently under construction for the installation of a screen to the already existing dam that will allow fish and debris to move above and over the surface of the screen. Therefore, conducting this experiment now is imperative to understanding how goldfish may affect the native plants of Pyramid Lake if goldfish become introduced through the fish screen in Truckee River. The timeline of events shows when lakes will be surveyed and when samples will be collected. So for starters, lakes will be surveyed at the beginning of the study to determine relative goldfish stocking densities that will be used in the lab experiments, and then also plant species richness and densities will be measured twice each month for a 5-month period. Goldfish densities will be measured using the mark and recapture technique using small T-tags and large hoop nets. To do this, we'll have to section off each of the lakes with large fish nets used for aquaculture in order to maintain a standardized volume of 3,000 gallons, so that would be an area of about 400 cubic feet. Large hoop nets will be placed in each of these sections in each lake in the morning of the first sampling day, and in the evening of that day, all fish will be removed and goldfish will be tagged with T-tags. Hoop nets will be replaced at the beginning of the second sampling day, and all goldfish, tagged and untagged, will be recorded. All netting locations will be accessed via kayak, and all nets will be removed after this portion of the study is complete. So, native plant species richness and densities will be measured by randomly placing 10 quadrats in each of the lake lakes that are 1 meter by 1 meter wide. 2 undergraduate students will scuba dive into the lakes to each of the quadrats to record data, and density of plants will be determined by counting the number of plants per species in each quadrat, and then species richness will be determined by counting the number of different native species per quadrat. Because goldfish begin spawning in April, and it is expected that they will become most active during this time, quadrat sampling will be repeated twice a month from April to August on the 1st and 15th of each month, giving us 100 quadrat samples per lake to analyze and compare. To calculate the stocking density of goldfish in each lake, the number of goldfish marked from the first sample day will be multiplied by the total number of goldfish from the second sampling day, and then this number will be divided by the number of marked recaptured goldfish to give an estimate of the total population size. The lakes will be sectioned off into the 401 cubic feet sections to standardize the water volume to 3,000 gallons, so the total number of fish estimated for 3,000 gallons of water using the mark and recapture technique will be used to simulate goldfish densities in the lab experiment on a smaller scale. To analyze the plant species richness and densities, the independent variable for the study is the presence of goldfish, and the dependent variables are the native plant density and species richness. In order to determine if native plant densities are altered by the presence of goldfish, we will first conduct a linear regression for each location over the course of 5 months to see if there is a significant difference in plant densities between the two lakes. That's what the figure on the bottom left is showing. Those are made-up numbers. We will then conduct a t-test to see if there are any significant differences in densities between study sites of goldfish, present and not present. And then native plant species richness will also be compared across study sites using a t-test to look for significant differences caused by the absence or presence of goldfish as well. All data analysis will be done using the statistical software R. So it's expected that we will find a high abundance of goldfish in Lake Tahoe and no goldfish in Pyramid Lake. If goldfish happen to be present in Pyramid Lake, it would likely be due to the construction of the dam on Truckee River. We also predict that native plant density and richness will be lower in Lake Tahoe than in Pyramid Lake due to the presence of goldfish in Lake Tahoe and absence of goldfish in Pyramid Lake. If this is not the case, it could be due to other non-native species interfering with the study sites or the lack of native species interfering with the study sites. This will be addressed by conducting lab studies to compare plant densities and species richness in aquariums with and without goldfish. This unexpected outcome could also be caused by invasive plant species, which could be addressed in future studies. If our hypothesis is supported with our field data, then we can conclude that goldfish have a negative impact on native plant species richness and densities.
We tested goldfish and native plant species together in a more controlled setting at Sierra Nevada University. We did this by setting up 4 55-gallon tanks, with 3 of the tanks containing goldfish and 1 containing no goldfish, which served as our control tank. Our control tank will determine if the decline of species richness and plant diversity is dependent on the presence of goldfish. The number of goldfish that go into the tanks will mirror the density that was found in Lake Tahoe by our first field survey. All tanks will be equipped with a sponge filter, sand, and native plants. As well as being kept at the same temperature and receiving the same light, they will all receive 4 native plant species, with 3 bunches of each species per tank will be planted, meaning 12 plants are in each tank total. All the necessary supplies and goldfish will be purchased from a local pet store, and the plants will be ordered from online vendors. For routine maintenance and data collection, the goldfish will be fed pellets once a day and have water changes weekly on their tanks to maintain water quality and maintain health of the water. During these water changes, the individual plants will be counted and the species noted. This will be used for the purpose of determining the plant species richness and densities. This will be done weekly for the 4 consecutive weeks. The independent variable for the study is the presence of goldfish, and the dependent variable is the plant's density and richnesses. The stats test will consist of a linear regression for each tank over the course of 30 days that will be used to see if native plant densities are altered by the presence of goldfish, and a t-test will look at the native plant species richness to see if there's a significant difference caused by the absence or presence of the goldfish. We predict that native plant densities and species richness will be lower in the tank where goldfish are present, and therefore will then be higher in the control tank where no goldfish are present. This would mirror the expected findings for the field study. Now, this lab work is important because if our field data shows that there was a decline when goldfish are present, but our lab shows the opposite, then could be another environmental factor or species that is causing the plant decline. If our hypothesis proves to be supported by both the lab and field data, this further proves the goldfish cause a direct decline in the native plant species. The budget shows the needed funds in a rough order, from people needed to field equipment, lab equipment, and finally travel. Everything is reasonably priced, and much of it could be locally sourced at the study site, so no shipping fees need to be added. The miscellaneous supplies refer to anything small that may have been looked over, such as water changing supplies, writing utensils, or etc.
This study is important for understanding the impacts of goldfish for the development of management and restoration plans in lakes around the world.